Hello everyone, I am honored to present our research, Genomic Adaptation of Clostridium perfringens to the Human Intestine. Clostridium perfringens is widely distributed in the environment and foods. Its heat-resistant spores can germinate and proliferate in undercooked foods, leading to potential infections. However, genomic investigation of C. perfringens isolates from the human population has been lacking. This study performed genomic analysis on C. perfringens from humans in China. There were regional and transregional transmissions of C. perfringens between individuals. The CPE gene was first identified on a PCW3-like and PCP13-like fusion plasmid. Genomic characteristics of C. perfringens were associated with host species. The average nucleotide identity, ANI, among the C. perfringens genomes ranged from 95% to 100%. The PCP13-like plasmids were identified in 20%, 39/195, of the isolates, while the PCW3-like plasmids were significantly more prevalent. Several ARGs were detected among the isolates. The major ARGs, Teta, P, TET-B, P, Q, and LNU, P, were detected among the C. perfringens isolates carrying different plasmid profiles. An ANI analysis revealed that some isolates exhibited an average nucleotide identity of greater than 99.99%. Pangenomics showed that these isolates displayed a similar genomic composition, suggesting a potential transmission event. C. perfringens strains can acquire external gene clusters during transmission, which may enhance their ability to incorporate and disseminate new genes in subsequent transmission processes. The PCP and TCP loci were both located on plasmid PCQ145 TCP-PCP, along with CPE, suggesting the fusion of PCW3-like and PCP13-like plasmids in CQ145. IS1469 and ISCPE2 were located at the junctions between the PCW3-like and PCP13-like plasmids, suggesting that these elements played a role in the formation of PCQ145 TCP-PCP. The C. perfringens strains in China were divided into 5 evolutionary clades, the human-derived strains clustered in clusters 2 and 3, while the animal-derived strains clustered in clusters 1 and 2. Several clinically important ARGs, including Optra, CFR, B, and CFR, C, were exclusively detected in animal-derived strains. The sheep, goat, and bovine-derived strains harbored more toxin genes than those from humans, suggesting potential differences in virulence among strains from different animal sources. The majority of the isolates were grouped closely in the maximum likelihood. No significant variances were observed in the accessory gene profiles of isolates from different countries, suggesting that the regional sources did not significantly influence the evolutionary relationship or genomic characteristics of C. perfringens. Small genetic differences were commonly identified between the strains with greater than 99.99% ANIs, highlighting the prevalence of transfer of gene clusters in C. perfringens strains during the transmission. The C. perfringens strains of the same animal origin clustered together in the phylogenetic tree, and a high proportion of the strains from humans, pigs, and chickens were found to share similar accessory gene profiles. A lower proportion of the human-derived strains were positive for PFOA and NAGI, while detection rates of COLA, NAGK, and NANI were significantly lower in pig-derived strains. In summary, this study comprehensively investigated the genomic characteristics of C. perfringens isolates from humans in China, as well as the population structure and pangenomic characteristics of C. perfringens worldwide. The findings offer novel insights into the transmission dynamics of C. perfringens strains, the localization of key virulencers, and a relationship between C. perfringens and host species. Toxin genes may play a crucial role in determining the host selectivity of C. perfringens, highlighting the importance of further research into the molecular mechanisms underlying this pathogen's virulence.